Hello, viewers. Welcome to Biodrunks. I'll be providing notes, PDF link in the description.
In last videos, we have learned about plasmids and vectors. So what are vectors? Vectors are vehicles, isn't it? To transport the DNA insert. Now, today we are going to learn about a bacteriophage vector where a bacteriophage is used as a vector. So what are they? First is what is a bacteriophage? So bacteriophage is a virus that is infecting a bacteria. Okay, so this is a bacteriophage. Okay, you might have remembered this is the head, here you have collarbone, these are the tails, and these are the arms, isn't it? And it has its own DNA. Oopsie, yeah. The other cell, the cell has its own chromosomal and plasmid DNA, and it infects it. So this is how it is, right? Now, this bacteriophage has two cycles. One is called as the lytic cycle and one is the lysogenic cycle. So what does it do in a lytic cycle? So when it has a lytic phase, here its viral functions are expressed fully. Okay, it's expressing its virus functions. Now, not only that, it has its viral proteins and DNA. Okay, that gets packed into its head or packed into viral particles. Okay, so it does that, and at the same time it lyses the host cell, lysis of host cell. So once it lyses the host cell, what happens? You have the release of virus particles. Okay, so this is how it happens. Now in the lysogenic phase, whereas in the lysogenic phase, what happens? When it gets inserted into the host cell, it gets inserted and remains inactive. So it remains inactive throughout the process. Okay. And then during some environmental conditions or tolerance or stress, any such conditions, it becomes active. It becomes active. Make it active. Okay. And once it is active, it again does what? It expresses its viral DNA, and then it produces its viral particles and lyses the cell, lyse of the cell. Easy? So both are nearly same, but here it's being dormant for a while. Here it just lytic cycle. It starts its own lysis, produces its own particles, whereas in lysogenic phase it remains inactive. Okay, so this is how the phage works, bacteriophage works, and we are using this bacteriophage. Okay, the reason we are using it as a vector. So bacteriophage, we use it as a vector because they are more efficient than plasmids. Okay, how? Because they can clone large DNA fragments. Okay, the first and foremost, they can clone large DNA fragments, and it is again easier to screen them. So easier to screen. Okay, these are the most important points because of which we are using it in place of plasmids. Now, these bacteriophages, their genome size is nearly 48 kilobase pairs. They have their own origin of replication. They have their genes for head and tail productions, proteins. Okay, and they have enzymes for DNA replication. Okay, and they have a most important factor called cause site. What is cause site? This cause site is a 12 base pair cohesive ends. So why do we use this cohesive ends? See, whenever you have to bind something, okay, so for example, this is it. Okay, you usually cut it this way is more effective to bind something, isn't it? This is a different fragment and this is a different fragment. This makes your bond very stronger. So this is why we use cohesive ends. Okay, they make our ligation very easy. Okay, so they allow the linear DNA molecule to form circular DNA by annealing the cohesive ends. And at the same time, during lytic cycle, the large number of when a large number of fake DNAs are matured, they help them with the packaging. Okay, so this is how we use coside and why it is very important. Okay, now what are the problems we face with the bacteriophage vectors? Okay, then we'll come to types. So the problems that are being faced with the bacteriophage vector is their size ranges between 38 kb to 52 kilo base pairs. So this means they can accommodate a DNA insert, but it can be only nearly 3 kilo base pairs of DNA insert. Okay, so what do we do is we have non-essential genes present in the phage. Okay, they are not used. So these non-essential genes, they are either deleted or they are replaced. So when in these two conditions, we can increase our DNA insert size. Okay, so we do this. Now, how do we do it? Because of these, the restriction, this is vectors, bacteriophage vector is of different types, insertion and replacement. First, let's do one thing. Let's draw our vectors first with their restriction sites. Okay, so we can understand it. If we do it together, we can understand it very easily. So restriction sites here I'm talking about, which is very important. So you can just check out my previous videos in which you have a detailed explanation about restriction enzymes. Okay, so here these are restriction sites of this particular vector. Okay, this is how I'm placing it. Great. Now I have let's start with insertion. So this is a non-essential region. Okay, this is a non-essential region. So this region is first deleted and then you insert your DNA insert. So this is the DNA insert. So can you see, once you deleted it, then you're inserting your DNA insert here. Okay, but in another case, we have something called supper segment, which is flanked with the restriction enzymes. So what do we do is we replace this particular fragment. We are replacing this particular fragment with the DNA insert. Okay, so here it comes out. So you are replacing the supper fragment with the DNA insert. This is called as replacement lambda vector. So this is how you have two types of vectors, insertion lambda vector and replacement lambda vector. Now let's talk about the advantages. So the advantages are it has storage is too good. Okay, storage of phase particles, and then the shelf life is infinite. Screening is very easy, and at the same time, transformation is also very easy. Okay, now different type of phage which we use here is M13 phage vector. Okay, so what does this M13 phage vector do? It is a single stranded. In it is single stranded. Where it is single stranded? It is single stranded in the phage. But when it enters the host cell, it becomes double stranded here. Okay, and different types of fakes are M13 are M13, MP8, M13, MP9, M13, MP1. Okay, see, very large inserts can be cloned. Okay, because the packaging does not depend on the genome cell. Not only that, the bacterial cells they infect, they are liable or viable and they form plaques, which makes your selection very easy. Like those forms forming the cakes are recombinants. So this was all about bacteriophage vectors. Now, because you know what is a bacteriophage and what is a plasmid vector, so we will be doing in the next video something about cosmid, phagmid, and plasmid. phasmid. Okay? Because we already did bacteriophage and plasmid vectors. So now we will see how they both, now separate vector, the separate vector, how if you can merge them both, what is it forming?
This was all about today's video. Hope you liked it. For more such interesting video, please like, share and subscribe to Biodrunk. Let's meet in the next video. Bye-bye.